Hello listeners and welcome to the Dr. GPCR podcast. Did you hear about the Dr. GPCR University? It's the ultimate learning platform within the Dr. GPCR ecosystem. Here you can expand your knowledge, connect with experts, and stay informed about the ever-evolving world of GPCR research. We offer both live and on-demand courses covering everything from GPCR pharmacology data analysis to drug discovery strategies. Our courses, like those that focus on using GraphPad Prism, help you master the pharmacology data analysis framework, navigate the complexities of GPCR research, and improve your work's quality, reliability, and efficiency. Join us and elevate your GPCR expertise today. And now let's dive into this episode.
Hello, everyone. This is Yamina from Dr. GPCR. We're back with another fantastic podcast episode. And this afternoon or this morning, wherever you are when you're listening to the podcast, I'm very excited to have with us Dr. Gabriele. And I'm going to mess up your name, Kockelkoren. There you go.
Yeah, that's good. Yeah.
Thank you. It's funny because I always ask our guests before how to pronounce their names. And I typically do mess it up whenever I'm doing the introduction. It's that little stress knowing that you're recording. Gabriele, thank you so much, Gabriele, for joining us. Very excited to have you on. You and I and the Dr. GPCR community has met you before because you've presented some of your work during one of our online events. So welcome back to the podcast.
Thank you so much. Yeah, I'm very happy to be here. It's great to see you again, and also to be able to share more about GPCRs. I've always appreciated Dr. GPCR. Also, when organizing the Early Career Symposium for scientists in Europe, that was great. So happy to be here.
Thank you so much for being here. So let's start at the beginning. I'm curious to hear about what do you do, who you are, and how did you end up working on GPCRs?
Yeah, so I am half Dutch and half Italian, which explains the funny combination of names. And so currently I'm actually at Stanford in the lab of Alice Ting. And I just moved here like 3.5 months ago. So that was quite a big change. Before this, I was in Copenhagen in Denmark. And so not only has the science changed, but also the weather has tremendously changed between Denmark and California. So in Alice's lab right now, I'm doing protein engineering to GPCRs because Alice's lab has this great history of developing proximity labeling tools. So they use these enzymes that catalyze a reaction, and then you can add a functional link like biotin to a protein of interest, and then you can resolve interactomes. So I thought that was very interesting in the context of GPCRs to try to find out new interaction partners. And now we've been developing this project where we will do interactomics for GPCRs or G proteins or arrestants, and then have subcellular precision while doing that. So especially there's this emerging paradigm that GPCRs can signal from the inside of cells. And so it will be interesting, can we map for these specific intracellular locations what these interaction partners are? And so that's what I'm doing now. And it actually, I got into contact with GPCRs during my PhD, not much earlier than that, because I started out in Delft in the Netherlands, and that's where I did my undergrad. And I did an undergrad in something called nanobiology. It was a fairly new program at the time. And the whole goal of the program was to create interdisciplinary scientists. So half of my courses were in theoretical physics, and the other half was molecular biology.
Wow.
Yeah.
Yeah,
exactly. So that was, but it combined for me exactly what I wanted, because we would look at very fundamental like processes in biology and then try to model them and then extract parameters that are relevant. I don't know, we were doing like in our first year projects about microtubule growth and catastrophe, and can you get the rates out of that? And that was very cool. And I remember while looking around in the department what kind of projects were out there, one of the things that struck the most to me was membrane biology. And to me, it was, so there was two parts of it that were fascinating. One was the fact that that the membrane is like this signaling hotspot. The fact that stuff gets like proteins get together, they form these complexes where you have a stimulus on the outside, and then you can propagate that kind of information carrying. I thought that was very fascinating, and how that would happen, and the fact that you can tune it, you can have specificity in it, so many different variables in it. And the other part is, and that I think is a bit underestimated sometimes, is that we think of the membrane as this kind of flat 2D sheet, but it's not. It's extremely rich. It can form these crazy shapes, really millions of different shapes. And you have these beautiful videos of like neutrophils swarming around, and you
I
can
love
see
it.
how, yeah, those are great. So for me, that was like that's where it all started. I thought the fact that they can make all these different shapes and that shape then also confers a function, that was crazy. And in some sense, it's also intuitive that shape confers function. If you think about, I don't know, if you look at the shape of a chair, the fact that it does confer that function, but in biology, you don't maybe always think about that, or not so much. And so that's what prompted me to go then to Denmark, to the group of Dimitrios Stamu. And Dimitri's group is using microscopy to study how heterogeneities at the plasma membrane of cells actually give rise to new functions. And as a model system, they were looking at G protein coupled receptors. And in Denmark, a lot of groups are actually looking at G protein coupled receptors, and that's because Novo Nordisk has a pretty important share in funding a lot of the research. And obviously, well, Novo Nordisk is very well known for its interest in G protein coupled receptor and insulin. And so that's how I actually got in touch with GPCRs for the first time.
I love it. I love it. And what a trajectory. I mean, theoretical physics.
Yeah. But it is. I mean, many things I don't use obviously anymore. We had also some quantum physics in there, and so that was fun to see. But but not for too long, because it was quite overwhelming. But I think the quantitative aspects still reflects in a lot of things that I do, and the way that I kind of try to think about receptor-ligand interactions or allosteric transmission to a G protein as you're an arrest. And I think seeing these as like rate-limiting processes and seeing these as dynamic, like models that you can dynamically model, that I think that's a very good way of looking at it.
I love it. So when before university, I'm assuming you had an inclination for science. Was it more physics, math, or maybe biology?
As a kid, surprisingly, I always said that I wanted to be a maths professor.
Okay.
Which my parents found that very weird, because I come from a family of musicians. Actually, my dad works in a symphony orchestra, and my mom has always played different instruments, and I don't do any of that really. That's because I have no sense of rhythm. So that did not really propagate to me, I guess. So I don't know. I thought numbers were very interesting, and that's what. But I thought when I was choosing an undergrad, I thought that just the numbers it was a bit too dry. I wanted something that was more tangible, and I think biology, because it's very visual, and it's that's why I like microscopy so much, for example, because it's so visual. I think the study that I chose was this great combination between having something quantitative and still having the numbers in there, but also being able to appreciate dynamics, things that move, things that I can see. So I think that got together quite well.
I love it. You mentioned, when you mentioned the microscopy aspects and the visual aspects, but also the fact that the membrane is not just this flat, static thing. I was at an Integrin meeting last year in January, and there was a lot of talk around mechanosensing and GPCRs, obviously, and integrins. And there was this physicist, actually, who was studying integrins. And it was this whole idea of understanding, for example, pressure. Or they had this tube, and they figured out how many cells does it take to migrate through, and how many you need a critical mass for the cells to be able to migrate through that tube. And the narrower the smaller the tube, the less cells you had. At some point, the whole thing fell apart, which I thought was really fantastic. And there's another group who was using Drosophila as a system, where they were literally pushing on the membrane to see how the membrane reacts to the physical force.
Yeah, I think it's always fascinating to think that physical force can be like an agonist. Like like we always think of agonists as like it has to be a small molecule, it has to be like a peptide or something more chemical in nature. But thinking about it as a force, that a force can be also a stimulus, like an activator receptor. I think much of the work that I've also done, I think really points towards that, is that just bending a membrane. If you bend a membrane, you are expanding one leaflet of the membrane and like compressing the other one. So you are immediately pressing or like pushing on that receptor, and so there must be allosteric modulation of that protein. And I think it's a bit underappreciated, but I think it's extremely fascinating.
I think it's underappreciated because I don't think we have all the tools that we need in order to study it. I think it's fascinating too. I mean, just the lipid composition of the membrane, changing that changes the receptor function or influences receptor function.
Yeah. So I think that was a lot of the work in my PhD when we started, it was around GPCRs. And one of the main goals that we first had is let's try to image them and see how they're localized on the plasma membrane, because there's been a lot of talk about these microdomains or nanodomains of GPCRs. And I think Davide Calibiro at Birmingham, they have done also a great amount of work in showing these nanodomains. And we had developed this technique to image the membrane in 3D so we could directly map where the receptors are. And there it was, that was actually again, a very visual, very beautiful project, because we immediately saw there were regions where the receptors were up concentrating and down concentrating, and it was clear there were spatial patterns. But that was only at the level of like density, so how many receptors are there? But that was not interrogating function yet. And so then we started adding conformational sensors and probes, and I think that's what I'm still kind of shocked about, is that we found that there are regions of the plasma membrane where receptors are kind of hyperactivated, much more active than in other regions. Some regions, we were looking at the basal state, for example, and we found receptors that were 20-fold more active than other receptors. And the fact that you have this segregation of receptors on the plasma membrane, I think can add so much functional value to the system in creating specifics and specific interactions. And that same held true when we added agonists. We saw even if you add, I don't know, we were working with beta-adrenergic receptors. So if we were adding isoproterenol or norepinephrine, we saw that even in that case, there were still receptors that were more active than receptors in other locations. And so it kind of prompted us to think that, you know, the efficacy of a ligand on a receptor is, maybe it's not just a single number, but it's a gradient of numbers that is mapped out in space, and some receptors are more active and have higher efficacy than other regions. And I think that still kind of boggles my mind.
Yeah, no, I agree with you. And what, unfortunately, in the cell-based assays, you measure the average of whatever you have, and it's also dependent on your system.
Exactly.
But being able to see it and to determine which region does what, not only from, as you mentioned, not only from a concentration of receptors, you know, as you were describing it, the image that came into mind is cells getting ready to move. So chemotaxis.
Yeah, exactly. The polarization.
Exactly. And I think it's fascinating because clearly it's a change in the membrane. The cell has a purpose. We're going that way.
Yes.
And then you need to reorganize everything. And to your point, even I have to change the way I think about cells, because I think about the cell, there's plasma membrane, there's your receptors, and inside the cell there's stuff floating, but it's not floating in there.
Yeah.
Everything is compartmentalized. Everything goes where it's supposed to go. It's this very intricate network.
I mean, to me, it's always fascinating that this even works. Like the fact that
we're here.
Yeah, we're here, which is great. But like and that we can observe it. I think I can see how imaging is gonna, I hope really in the future that imaging is gonna make a big breakthrough, and that we can really study receptors in this native context, in a more native context at least, and really see interactions with agonists, see how they're organized, how does this promote reorganization at a bigger scale, because at the end of the day, like earlier you were talking about mechanosensation, for example, more in an organism kind of view, but really integrating all these levels from like a single receptor to a domain of receptors to a whole cell, etc. That integration is still also kind of missing, and we don't know the missing links there.
So what, speaking of this, let's say, ideal advancement in the field to get us to be able to look at this and get a sense of these events, what kind of tools do you think we need to get there?
Yeah, so I think that's a tough, like, ideally speaking, I think but we are currently kind of focused on a very receptor-centric view. And while we've had like amazing insights from crystallography and from cryo-EM, and they've really guided our drug discovery kind of pathways through like structure-guided design, we're still kind of, these receptors at the end of the day are still taken out of their native context and put into something that is definitely less native. And we don't know how that translates. Like, we don't know whether what holds for a receptor in a certain detergent actually holds in a much more complex and organized plasma membrane. So I think if we really want to move forward, especially in terms of drug design, I think it would be great if we can develop tools that assess the ligand action at a kind of molecular level, but in living cells, in intact cells. Like, it would be great if you could do live cell NMR. Can you map, for example, changes in transmembrane helix 6 or in live cells? Or can you do that with fluorescence microscopy? Can we really look at conformational states, biasing in living cells at the receptor level before even moving outwards towards all the different cascades, kind of that biochemical cascades that come after that? I think trying to understand these receptors in a more native context is definitely pretty key to advancing the field.
Absolutely. I think the first thing that comes to mind from a tool perspective is antibodies, specific antibodies to the receptor, which is a challenge.
Yeah, that's extremely. I think we're thinking also here in the lab with Alice. We're thinking about nanobodies that can target the extracellular side of receptors, so that we can kind of start modulating receptors when they are natively expressed, instead of trying to work with them when you overexpress them. Yeah. And how does that translate? I mean, it's very well known that overexpression messes up the system in multiple ways. Even when doing, for example, just measuring EC50s, you see shifts in these curves. So I think it is very valuable to create tools that allow you to interrogate how a native receptor is interacting with hopefully minimal perturbation. I think to that end, that's part of why I joined this lab, was that I think it's interesting that we know that there are arrestins, we know that there are G proteins, and quite a lot of interactors are quite well characterized, but there must be missing pieces and missing molecular interactions. And what I like about Alice's lab is that they're trying to develop these tools to kind of get in without perturbing the system too much, can I get the interaction partners of a GPCR or of an arrestin in a specific context? So if I don't know, it would be a dream to like find not a new G protein, but like I don't know, another protein that is also recruited to the plasma membrane, has its own kind of dynamic. Maybe it co-segregates together with the receptor into the endosome, or it would be very, to me, that's very fascinating if we could find these kind of molecules that contribute to the specificity of signaling.
Yeah, and people are working on these, and I think if I remember correctly, Joanne Trejo's lab has been very focused on such work. And yes, because when you think about GPCRs, especially from a drug discovery perspective, people think about G proteins, beta arrestin. That's it.
Yeah, it stops there. Yeah.
The purpose is different, so I do understand why, but I think to get a better sense as to what the receptors do, a holistic view is needed.
Yeah. I mean, I'm very much focused, I think, personally biased to trying to get molecular mechanisms, or trying to understand why does it work the way it does. Yeah. And so I do wonder whether we're missing parts of or like which interaction partners are we missing that will fill up this picture and give us the missing puzzle piece.
No, I agree. I absolutely agree. All right, what is your favorite GPCR? Because I have to ask this.
Yeah. So I started out by working on the adrenergic receptors, which was great because these receptors have so many different ligands and they're so well characterized. But then I started working on a receptor that I found even more fascinating, which was the glucagon-like peptide 1 receptor. So these GLP-1R, the incretin receptors. And I think there's multiple reasons why I think that's a very fascinating receptor. The foremost reason was that when we started using that receptor in my lab, we found that we were mapping the location of the receptor at the plasma membrane, and we found it had these very distinct domains. And then when we added GLP-1, which is the native ligand, we saw a complete redistribution of where the receptors were locating. And first off, we did not expect that. And secondly, it really suggested that changing the conformation of the receptor was altering its preference for certain lipids, for certain curvatures, for certain tension, any kind of property of that plasma membrane. And so that was really a breakthrough moment, kind of, in my PhD, which was very unexpected, but extremely fascinating. And then I think the other reason that I'm so interested in GLP-1R is, well, nowadays, a lot of people are interested in GLP-1R.
Everybody is, yeah.
But I think there's like an interesting history to GLP-1R and to its discovery. I think in the 80s, GLP-1 was discovered for the first time, but actually it took a long time before we got from using GLP-1 as an actual drug nowadays, because the biggest problem with GLP-1 is that the two N-terminal amino acids get cleaved off by a protease, and so it had a very short lifetime in humans, and it actually took a long time before GLP-1 could be used, and nowadays we have these ligands like semaglutide or liraglutide that have look a bit like GLP-1, but somehow they produce very different and they are very efficient in terms of agonistic properties. And so to me, it's interesting to understand how is it possible, or like, first of all, why do these peptides work as they do? But also, for example, why do dual agonists that target GLP-1R and, for example, the GIPR, you have these like tirzepatide, how is it possible that dual agonism or like triple agonism can give you very different effects or very new effects or reduce side effects, right?
Yeah.
So I would really like to understand this from a more molecular perspective, even though we do already know a lot about GLP-1R. I mean, there's like 40 years of research, and but
great. No, I was taking notes because just as a side note, I'm going to ask you what should be the title of our conversation at the end. But my team has been asking me to actually take notes, and we're going to have short videos. And one of the short videos is, what is your favorite GPCR? So I was trying to come up with a title, and our team internally who produces these videos, they're not GPCR people. They're not scientists. Yeah. So one time it was funny because one time we used to at the time we used Zoom, and we had the AI transcript, and instead of transcribing adhesion GPCRs, Zoom rewrote it and it became Asian GPCRs. And we were looking at it, and the team was like, Yamina, I think something is not right there. So that's why I was taking notes. But to your point, I think nowadays everybody has heard about GLP and GIP and all of these drugs. And if I remember correctly, initially GLP was discovered at Rockefeller University.
Yes.
Yeah. At the time. And yes, there's a whole slew of questions. There's also a lot of research that has been done, so we're understanding better and better. A couple of years ago, way before the frenzy we're seeing today, I was at the pediatrician's office Actually, no, it was at the doctor's office, and there was this poster, and it was obviously semaglutide poster. And typically, when I think about science, there's always a GPCR involved in it. So my first thought at the time was like, "Ha, that's a GPCR."
Yeah.
There you go.
Yeah.
But I think right nowadays everybody is interested in it, and everybody has heard, even the general public, about GLP-1.
Yeah. I think it's also interesting for the general public because it brings up some moral questions about how you should be using this, and should everybody be using this, and should it be a substitute for, I don't know, exercise, or should I also think in that sense it's a GPCR that brings up a lot of questions, not only from a scientific point of view and out of like curiosity, but also a societal kind of point of view.
That's a can of worms, and I could go deep down into that can of worms. It's like, I think nothing replaces exercise and sleep and good food.
Yeah.
I think in general, I was listening to a podcast, I can't remember, but the person was mentioning that in their opinion, and I agree with them, drugs are great, especially because it's an acute, you need help, so you need a drug. Long term, nobody knows what's going to happen, and what taking that drug long term how will that affect you.
Yeah, no. So I actually have a personal reason for appreciating GLP-1R, because I am diabetic myself. So because of that, I think, and because I know how it is to have diabetes, I think for me it gives me it is an extra drive to try to understand how this works, and to create a drug, or create a treatment that is actually viable and can better people's lives, including my own.
Of course.
Yeah. So I have multiple reasons kind of to be excited about this. But actually, the reason that we worked on GLP-1R was, again, because it was an interesting system to work on. It was not. We were really truly interested in the biophysics, and I think this was still before like Ozempic got into the market. Yeah.
I think I agree. I remember it was way before the hype.
Yeah.
And the interest we're seeing today. All right. So can I ask, what made you move from Europe to the US for a postdoc?
Yeah, of course. So during my PhD in Denmark, it's actually a very great opportunity. You are, let's say, obliged, it's mandatory to have a research experience abroad. And my PI, Dimitri, he had a collaborator here at Berkeley. And so I went to Berkeley for 3 months and basically did single molecule imaging of GPCRs. And so being in Berkeley, I got to appreciate the Bay Area. I got to appreciate the sun that the Bay Area has. And also, I think what I like about the Bay Area specifically is that there are 3 universities here that are close together. So you have Berkeley, UCSF, and Stanford. And there's actually quite a lot of collaboration between them, and quite a lot of initiatives that try to get them together. So I think in that sense, it's like a scientific hub, and it's extremely interesting to experience it also like that. To experience, you meet so many people that are scientists. I take the train every day, for example, and most of the people that I talk to are postdocs. And so you get to meet a lot of people that do a lot of different stuff, and I think that's very fascinating to me. The other thing is, I wanted a change of environment. I wanted to, I think Europe has its, or not even, I don't even want to say Europe, but like Denmark or the Netherlands or Sweden, for example, have a certain way of doing science, and of how do you talk with professors, or how do you talk to PhD students. Denmark, for example, is a country that really relies on no hierarchy. And so it's a bit more of a flat level, which is very interesting. But the US has its own culture and its own way of talking about science, its own way of writing and presenting science. And I thought it was important for me that if I want to stay in science, and if I want to be able to talk to everyone in science, I should also be able to communicate with everyone in science. So I thought that this was a great opportunity also for that, just to get to know new people that do science in a different way, and really to just widen, in many ways, just widen my own horizon. And I think I'm getting a great opportunity at that kind of year. So those were the main reasons to move. And also, I feel I'm young now, so I can do this. I can
Yes.
go, and I didn't feel like settling down yet. Yeah.
I think it's a great opportunity. To your point, it's a great opportunity. I think the whole Bay Area is just fantastic, and
Yeah.
Stanford is Stanford at the end of the
Yeah,
day.
yeah. I mean, the first day that I'm very Dutch in the sense that I take my bike everywhere. And so I get here by bike, and I started biking up campus, and you drive up Palm Drive, and you have all these palm trees. And it's like, the first day it was really like magical. And it's like, oh, I work here. It was very, very beautiful. And I mean, I do enjoy the sun, of course, and California has a lot of good food to offer. So there's a lot of good things that come together here. And so I'm actually happy that I made the move.
I'm glad.
Yeah.
How did you, for those who are maybe finishing up their PhD or looking at the next step in their career, if you can give us a little sense as to your thought process, and how did you, more in a tactical way, of how did you go about finding this postdoc opportunity? And
Yeah. So I knew I wanted to go to the Bay Area because I had been here before, and I thought, and obviously this is a very good place to be in. So that restricted the search but I mean, it restricted the search, but still, you still have 3 locations. So I was looking for Berkeley, UCSF, or Stanford. And basically, I already had in mind that I wanted to continue working with GPCRs. So then I started compiling lists of PIs that were either that I thought could be interested in working with GPCRs. They didn't per se have to be GPCR experts, or that were actually GPCR experts. So I had a list of people. I think my advice would be to start early with applying. I just sent an email and asked, and basically I said, like, I'm interested in a postdoc opportunity, and could we have a meeting? That was, and I started, I think, a bit more than a year before graduating from my PhD. I think it's important to start early because there's many things happening during your last year of your PhD, and things never take the time that you want them to take. So between writing your thesis, trying to prepare for a move, trying to prepare your visa, especially if you have to get a visa, these kind of things all take a lot of time. And I felt that I wanted to be sure that everything was like gonna pan out. And so I felt, okay, better start early than starting late. That also meant that I could apply for fellowships and these kind of things. So that helped me a lot. And also, what I really liked about working together with Alice is that from the start, we already started planning projects. So and thinking, we had brainstorm sessions. What do I want to do? What is interesting to me? What is interesting to her? And how do we bring this together? So I think that was very important, because then when I arrived here in the lab, in the second week, I basically already had my first cloned constructs, and I kind of started going because we had a very clear view on what I wanted to do, which helped me a lot.
I love it. I love the start, the starting to plan early enough, so that you know what you have to do once you settle. And it's rare, I want to say, in science in general. Anyone I interview, even PIs, when I say, well, did you plan, and how did you plan it? And I don't remember anyone who said, yes, I planned it ahead, and it just, things kind of happened. Obviously, you need to start early enough, but I think this is really fantastic on that front.
Yeah, if I could also add to that, is that what I also did is I went to a few conferences in that last year of my PhD, not just to find a position, but also just to talk to other professors and kind of ask them, like, oh, I'm interested in this. Is your lab, or are other labs? Do you think there are other labs that would be interested in doing this, and where I could apply? Or do you know these people? And kind of try to establish also connections on that side, because I mean, obviously I did that. I talked with my own PI, with Dimitri, and he gave me loads of advice, and he was very supportive in letting me go to basically any conference I wanted to. So that was very good. But it's also good to like see different views. I mean, Dimitri also doesn't know everyone in the world, so it's good to talk with other people and get other views on how to find a postdoc.
Yeah, I think you made not only how to find a postdoc, how to find a next job, how to find a collaborator. I think you made a fantastic point, and I think it comes up during almost each and every podcast episode, because I think it's important to keep an open mind to be able to talk to people. And people are afraid of this word networking, and before I started networking, I was afraid of it too during my PhD and early on in the postdoc. But what I realized is just being human and talking to people and just asking them questions.
Yeah, I think maybe the word networking is overvalued. I think at the end of the day, it's just talking.
Exactly.
It's just trying, like, being nice, being respectful, and just trying to learn about the other. And it's not more than that. Like it doesn't have to be more than that, really.
Exactly, because people, as humans, we have to label things.
Yeah.
And it is true that this word sounds like such a difficult thing to do. But in reality, you just talk to people. We are networking right now.
Yeah, exactly. And I like it, because then I mean, I hope to see you at the next conference, kind of, and you can talk back about previous conferences together, and you can. Exactly. I mean, you also create more personal relationships next to the scientific ones.
Yeah. And again, another great piece of what you just said. I think it's important because you do create the personal relationship, and this way you get to find out who are the people around you who you can not only work with, but you actually like working with them.
Yeah.
So they can call you up, you can call them up, and they can make introductions, you can make introductions, and it's actually quite valuable. And it's really being connected.
Yeah, absolutely.
I don't remember who it was, but I think it was Silvio Goodkin, right around the time where COVID started, and we had a Zoom call at the time, and he didn't talk. He said, we're not social distancing, we're physical distancing, because we're social people. And we're not socially distanced, we're just physically distanced in that front.
I like that.
I thought it was really nice too.
Yeah.
Speaking of COVID, how much did COVID affect your PhD, the end of it or the middle of it?
It was kind of in the middle. Towards the start, early middle, I would say. So I was lucky to be in Denmark, to be honest, because our university, I think, shut down for 3 weeks, and so that's not too long. And we were able to, like, it completely shut down, and then we were able to go in sometimes if we wanted to take care of cells or this kind of stuff, but not really long. And then Denmark implemented this policy where the whole country got PCR tested every 72 hours.
Wow.
So we just went to work because you get tested every 72 hours. And as soon as you had a sneeze or anything, people were very considerate, and everybody just went, like, people just go home and they take care of themselves. They go tested again. If it's negative, you go back to work. Otherwise not. So I was very lucky because the shutdown was relatively short. Everybody was being tested all the time. So in some sense, society could continue as it was. And I think what helped me a lot was that I had a lot of international colleagues in the group. And so we were kind of stuck in the same boat in that sense. So now you create your own little bubble, and it actually got us even closer because of that. Because you could only hang out with so many people. And we were seeing each other in the lab anyways. So then we would just hang out with each other as friends. So
I like that.
Yeah.
You formed your own little bubble there.
Exactly.
So we're all together. But how many people live in Denmark to be able to PCR test everybody every 72 hours?
There's like 5 to 6 million people, so it's not too bad, actually. and it's amazing that they were able to do that. Actually, I remember it was like I remember it because it was every Wednesday, every Sunday. It's like,
Wow.
and we were standing in these like just long lines in the cold, just waiting for the test. And it was like I don't know, like a typical Sunday morning appointment. Like instead of going to mass, you went to you went to get your COVID test.
That's very, I didn't know that. That is very impressive.
Yeah, it was in that sense lucky, I would say, to be there at that time.
Absolutely. All right, top 3 aha moments you had as a scientist that shaped your trajectory.
Yeah, so top 3 aha moments. I can either think of like actually scientific things, but also of more human things that happened.
Whatever works. It can be 5.
So like from a human point of view, or like from a more learning myself and learning to be a scientist, I think what I learned is that you will always feel like you know nothing, and that is completely fine. And I think that is actually why you stay curious, and why I mean, right now in Alice's lab, we're doing a lot of things that I've never seen before, never done before. There's a lot of chemistry happening that I've never seen before because I'm not a chemist. And I think it's actually interesting to, after your PhD, because when you're a PhD and then you're about to finish, you're actually quite senior typically in the lab. And then you start your postdoc and you kind of like have this whole moment of learning again. And it's very refreshing, and it's very challenging as well. But you kind of realize how little you know, and it's good. I think it's a good thing to be reminded of, that you know very little, and it also pushes me to learn more.
Yeah, I feel like it's a very humbling thing as well.
Yeah, very much, very, very much. And another thing that I really, and I think this is part of the COVID story, is I cannot underappreciate like colleagues. I think having good colleagues and having colleagues that you can rely and trust on and that are your friends is essential part of the lab, and essential part of what motivates me also to come to lab. And I think for nobody can say that their PhD was easy or that it was like a walk in the park. There will always be moments that are extremely frustrating for one reason or another. And I think being able to share that and having a little community, your own little community, even if it's one person, but as long as you have that, I think that helps a lot in getting through the PhD, but also getting through your job.
Yeah. No, I agree. And I absolutely agree. I think I don't know anyone whose PhD was a walk in the park.
No, yeah, exactly.
It comes with ups and downs, but I think that's what allows you to build some of that thick skin or thicker skin.
Yeah.
To then move forward. I love it.
Yeah. I think another moment that, or it's not per se a moment, but it's something that I realized by going to conferences and by going to talks, is that you can be a extremely good scientist, but if you're not able to talk about it, or if you're not able to convey your ideas, it's just not going to work. And I think presenting and being able to narrate your science is just as important as being able to do good science. And it's just so nice to go to a talk, and even if it's about something that you don't know anything about, but just have the feeling that you got enthusiastic about it just by hearing someone else talk about it. And I think that's something I think that's one of the most fun things about going to good seminars, is getting enthusiastic about a topic that's not yours.
No, I agree. And to that end, I feel like that comes through. That's an art in itself, to be able to tell a story, not just say we pipetted this and we did this.
Yeah,
and that's the thing. But at the same time, I think it goes back also to inspiring others. I mean, having that good biology teacher.
Exactly.
That told you these stories and got you interested. Any other teacher for in itself. As a PhD student for a long time, I thought I thought the problem was with me, because I would go to some seminars and be like, why are we even talking? Why did you even work on this? I'm not understanding anything. And that's, after a while, I realized that it's a matter of who is presenting and how that person is presenting.
Oh yeah.
If they're a good speaker, then you're gonna get more involved. And it's, I don't know about you, but even to this day, it's one thing to put the experiments together, to run the experiments, to get the data, but then once you have it, you've analyzed it, to put together a deck, a slide deck that tells a story, and it's not a cut and dry data, is an extra set of work.
Oh yeah, it's for me, or like at least how I see it is, I just have to sit down and spend like an entire day almost just like thinking, like how does this fit in this context? How can I tell this? How does it interest people? Or how do these things follow up on each other in a kind of, I mean, it has to be like a book. It has to be like you're reading an actual story, and most people don't know anything about what you're doing. So it
exactly. Yeah. Exactly. And then you have to also think about the who cares and why should people care questions. And I feel like, unfortunately, it's not emphasized enough during the training.
I agree. Yeah.
The story part, the making a case for something. Just as managing people or leadership during a postdoc or PhD, you don't teach that. You kind of end up having a lab, and then you navigate through it, and with time you get better, but at what cost?
Yeah, exactly. No, I realized that's what I kind of what I'm liking a lot about Stanford here is that I get a lot of emails with like different workshops by professional people that, what they do is teach people, how do you write, how do you present yourself, how do you pitch your work in a minute, or which I think is, so I'm planning on like taking different of these courses, because I think it's extremely valuable. It's just as valuable as me being able to pipette the right reagents together in the tube. It's just as valuable to make you enthusiastic about me pipetting reagents into a tube.
Exactly, and you explain to people why that is important and what you've learned. Why should people care?
Exactly. Yeah, absolutely.
So, no, agreed. Absolutely agreed. All right, 2 more questions.
Yes.
Any additional advice, because we've talked a little bit about it, to scientists who aspire to contribute to the field? I'm thinking here, the summer intern, the master's student, the PhD student, fellow postdocs. What have you learned? I
think something that I keep in mind a lot is that I think this is relevant to any field, but also to GPCRs, is interdisciplinarity is key. I think GPCRs have for a long time been studied with very using a lot of biochemical techniques, and I think it would be great if we can combine more and more techniques together in order to study GPCRs. So I don't think currently, for example, like a lot of super resolution microscopy, for example, is being done on GPCRs. That's actually quite a limited field. I think it's interesting to be able to pick techniques from one lab or pick certain methodologies and trying to combine them with GPCRs to answer new questions. And the best is if I like the best questions are the ones that are the most basic ones or the most fundamental ones. And so I mean, they're also the toughest ones, but that's kind of what makes them exciting. So I think if you I don't know, do your master's project or your PhD in a certain field, try to look for something that is very different, and try to apply to GPCRs. Try to think, what kind of interesting questions can we answer about a GPCR that were not previously attainable by using other techniques that we've had. So I think trying to find this interdisciplinary approach is for me the most exciting thing.
I love it. And you're right. But a lot of guests, when I ask them this question, they say, well, read broadly, read about what others are doing in their fields. That is a great piece of advice. It's a difficult one, but it goes back to being able to look at what others are doing in different, adjacent fields, and then try and bring that in to the field.
Yeah, and I think especially at the end of like a master's or a PhD, is when you are looking for like a new challenge. I think it's good, or like, yeah, I think it's best to kind of like try to step away a bit from what you're used to and try to take a deep dive into something new. While the other pool is still close by, but you
Exactly.
still wanna jump in a different pool. Yeah,
But it's difficult at the same time because,
absolutely.
you've spent such a long time developing your skills, 4 or 5 years, depending on where you're doing your PhD,
Yeah.
and then completely switching fields. Wow, what a jump.
Yeah. It's a trial by fire, but uh,
Absolutely. It's the deep end of the pool for
yeah.
sure.
But I think I hope I can say I hope it's gonna be rewarding. Yeah,
Yeah, I mean, these are high risk, high reward type of situations, and
yeah.
no matter what, you end up learning something, whether it doesn't work, or I
No,
hate it, or I love
yeah.
it. I think it's all worth giving it a shot.
I think it just depends on what you define as the reward, really. And
Yeah,
I think I mean, when I came here, I felt I would just want to learn new stuff, and if that's what I'm gonna get out of this, then that's great.
yeah, yeah. What next? Actually, I have another question before we move
Yeah.
on to the last one. What
Yes.
next after your postdoc? I know you're in it for 3 months
Yeah.
now, but considering
Ooh,
your forward thinking,
yeah.
I'm curious to hear.
So I'm for now my mind is set to stay in academia, and I think I will never be able to leave GPCRs because it's like it's too fast. It's such complex little machines. It's amazing what they can do. I would like. I think I would like also for personal reasons to move back to Europe, and I think maybe the Netherlands, but I also would not exclude Denmark because my time in Copenhagen was great. I love Denmark. I loved Copenhagen, and there's also many other places in Europe actually that I think are very attractive for doing science. So my plan would be to. I think it would be great to at some point move back and find an academic position in Europe. Um, in terms of the science that I would like to do, well, obviously GPCRs, but I think I would like to combine the fact that we use in the lab here with Alice, we're using protein engineering, but also a lot of like high throughput bulk types of measurements. And I would like to, it's very interesting to me that in a lot of bulk measurements, there's a lot of heterogeneity. But that heterogeneity is kind of concealed in means and averages and standard, like, we plot it as a bar graph, put an average and a standard deviation of it, and we say, okay, that's it. But I think there's a lot of information concealed that kind of goes unnoticed. And whereas when I was doing my PhD, I was especially looking at that heterogeneity because that was the interesting part, the fact that there was heterogeneity in the system. So being somehow able to combine this or bridge this, understanding heterogeneity also in larger systems, not only at the nanoscale, using these bulk types of approaches, I don't know, like FACS or, plus combined with protein engineering, I think that would be interesting to me. Yeah.
I agree, and it's interesting when you mentioned the bar graph and the average and standard error, and it's done. And when you're having technical replicates,
Yeah.
then you can look at them, and sometimes you have two there, one here, and then the fourth one somewhere else, and you might be one. Obviously, you don't have necessarily the time to look at this, but it makes you wonder, why is it the way it is? Is this reproducible? And you mentioned FACS. I haven't gotten a chance to use it, but I've always been fascinated by the single cell FACS, where
Yeah, me too.
you're sorting and imaging at the same time, and then so you get a sense of what each cell does. That also generates a lot of data. So I think adding in computational biology in there.
Yeah, or omics, like that.
Yes.
Yeah, absolutely. So like, I honestly, I have also not used FACS, but I've been seeing a lot of FACS data lately. So every time I look at it, I'm like, this is full of information.
It is. It is. I used FACS for a long time. I really love flow cytometry, but I think it's also somewhat not that of an exact. So let me take 3 steps back before I get emails from people saying that's not true. Okay, just to kind of clarify, you take your cells, whether they're endogenous expressing your GPCR or not, you label them hopefully with a good antibody, and then you run the cells. So you're acquiring, and you have this you can see the dots. Each dot is a cell. And when you're running your negative control versus your positive control versus your sample, where you put your threshold is, I think, a little bit, you're looking at it, but it's very hard and it becomes a little bit arbitrary. So you have to play around with it. So your percentages at the end.
All right. Yeah.
Yeah. So it's not that exact. It's not exact enough to my taste compared to, you're measuring and you have a number.
Yes. Yeah. Absolutely.
That's what the plate reader gives you, and that's the number.
Yeah. There's a human component in the
exactly
in the extraction. Yeah.
Exactly.
Yeah.
All right. Last question.
Yes.
What should we title this episode?
Oh, so I think what I always hope is that, in terms of like what people learn from my work, or like is that where a GPCR is matters. And so if I would title anything, even almost my PhD thesis, I would say GPCR location matters, or why GPCR location matters. I think it's important to realize that it's not a homogeneous system. Cells are by definition out of equilibrium, and there is variation, there is heterogeneity in GPCR location, and that confers function. So
yeah, and to your point, that's actually location, timing, speed at which things happen. All of this matters. It's interesting because we were talking 2 questions ago, and one thing you mentioned stuck with me is the fact that interdisciplinarity is key to move the field forward. So now I'm torn because I like both titles.
Both are fine with me, to be honest. But because I feel strongly about both of them.
I wonder if we can combine both in some way, shape, or form. But I think to your point, I think being interdisciplinary, and then also being able to actually, the thought I have is you need to combine tools from other fields in order to understand the GPCR location and to then understand why it actually matters. So there you go.
Yes.
All combined. Fantastic. Thank you so much, Gib. Thank you so much, Gabriele. I really enjoyed our conversation.
Me too, very much so. Thank you.
Thank you. Don't go anywhere.
I need a favor. If you enjoyed this episode, please click the like button. And if you like the podcast, consider subscribing to our YouTube channel. Many of you come back to watch our videos but aren't subscribers yet. One click goes a long way. All the information about Dr. GPCR is in the description. Find all our fantastic features and everything you can benefit from becoming a premium member. And while you're on it, check the Dr. GPCR University courses and share them with your network, colleagues, and friends. We also want to hear from you. Is there anything missing from our ecosystem? Can we make it better? Email yamina@drgpcr.org or email our team at hello@drgpcr.org. Thank you so much for joining us in this podcast episode. If you're still there, we also want to take a minute to thank our team members: Yamina, Attila, Montserrat, Cam, John, Yatsu, Mark, Ivana, and Andreina. If you like our podcast, you can leave us a 5-star review on Apple Podcast. Until next time, and stay safe.